In molecular biology, polymerase chain reaction or PCR is a technology that is used to make multiple copies of a segment of DNA. Kary B. Mullis developed the polymerase chain reaction in 1983. Earlier methods of obtaining multiple copies of a specific sequence of DNA were time-consuming and expensive. In contrast, PCR can create millions of copies of a single gene or any specific piece of DNA quickly in a test tube. PCR is very specific. The targeted DNA sequence can be less than 1 part in a million of the total DNA sample. This means that a single gene or smaller piece of DNA, among all the human genes, can be amplified and copied using PCR. PCR takes its name from DNA polymerase, the enzyme that carries out DNA replication in a cell. It is considered a chain reaction, because DNA polymerase will carry out replication, over and over again, until there are millions of copies of the desired DNA. A single strand of DNA is copied into 2 strands. 2 strands are then copied into 4 strands and the process continues, until there are millions of copies of a single DNA strand. Before carrying out PCR, primers must be available. These primers are sequences of about 20 bases, that are complementary to the bases, on either side of the target DNA. The primers are needed, because DNA polymerase does not start the replication process, it only continues or extends the process. After the primers bind by complementary base bearing to the DNA strand, DNA polymerase copies the target DNA. A basic PCR setup requires several components and reagents. These components include DNA template, that contains the DNA region, target, to amplify. Primers, Taq polymerase, and deoxynucleoside triphosphates. The PCR is commonly carried out in a reaction volume of 10 to 200 microliter in small reaction tubes, in a thermocycler. A thermocycler, also called thermocycler, is an instrument that employs precise temperature control and rapid temperature changes to conduct the polymerase chain reaction, PCR. DNA polymerase used in PCR is temperature insensitive and thermostable enzyme, extracted from the bacterium Thermus Aquaticus, which lives in hot springs. Commonly this enzyme is also known as Taq polymerase. It can withstand high temperatures, which is used to separate double-stranded DNA. Therefore replication need not be interrupted by the need to add more enzyme. The whole process can be divided into following 3 steps. First step is the separation of 2 DNA strands. At very high temperature, hydrogen bonds between 2 strands break and the 2 strands are separated from each other. Second step is annealing. In this step, the temperature is lowered, to allow the primers to bind the DNA. In the next step, the temperature is again raised, to allow the DNA polymerase enzyme to synthesize new strands of complementary DNA from free nucleotides in solution. PCR does not replace gene cloning, which is still used whenever a large quantity of gene or protein product is needed.